Welcome to this explainer, where we are going to completely unpack the true nature of cholesterol. We're talking everything from its deep, complex biochemical pathways to real-world clinical treatments. Whether you're a food technologist analyzing lipid profiles, a toxicologist studying pharmacological pathways, a chemist, a clinical dietitian crafting interventions, or a doctor managing patient care, this is for you. We're going to bridge that gap between hard biochemistry and highly practical, actionable insights. Let's dive right in.
Is cholesterol really the enemy? It's a completely valid question, right? For decades, cholesterol has been totally vilified in popular health media. And yes, while hypercholesterolaemia, having too much of it in your blood, is undeniably a major risk factor for coronary heart disease, the molecule itself? Absolutely not the enemy. In fact, cholesterol is a literally vital building block for our bodies. It maintains the structural integrity of every single cell membrane you have. It's required to form the myelin sheath that insulates our nerves. Plus, specialized brain cells rely on it to build the synapses that let our neurons communicate. Without cholesterol, we couldn't synthesize vitamin D, we couldn't digest fats, and we wouldn't produce essential steroid hormones.
Here is our roadmap for today. 1. The core of cholesterol. 2. Meet the transporters. 3. Assessing the risks. 4. Dietary management strategies. And 5. Medical and alternative treatments.
Section 1. The core of cholesterol.
We really need to understand where this vital molecule comes from and how our body actually builds it from scratch. Think about this for a second. An average 150-pound adult walking around right now contains about 140 to 145 grams of cholesterol. But here's the wild part. The vast majority of this isn't just freely floating in the blood, and most of it actually didn't come from the eggs or cheese they had for breakfast. Your blood only contains about 8% of the body's total cholesterol. Most of it is synthesized de novo, meaning built entirely from scratch, primarily in your liver and intestines. Your body has this incredible feedback mechanism to ensure it always has exactly enough of this crucial compound.
When we trace the biosynthesis pathway, we find that all the carbon atoms in cholesterol originate from acetyl-CoA, which is just a basic metabolite of carbohydrates, fats, and proteins. But the absolute crucial choke point happens at a very specific rate-limiting step. The enzyme HMG-CoA reductase reduces HMG-CoA to valvulinic acid. Now, why does this matter to us? Because this specific enzyme is the master control switch for cholesterol production in the entire body. If we can control this enzyme, we can control blood cholesterol levels. Keep that in mind because it's going to be incredibly important when we talk about pharmacology later on.
Section 2. Meet the transporters.
Because lipids like cholesterol and triglycerides are essentially fats, they aren't water-soluble. They can't just dissolve into our water-based bloodstream. They need vehicles to travel through the bloodstream highway, and that brings us to the lipoproteins. We can map out our body's lipid transportation system pretty cleanly. Think of chylomicrons as your immediate post-meal delivery trucks, moving dietary fats directly from your intestine. VLDLs are your liver's delivery trucks, shipping out the triglycerides it made itself. Then we have LDLs, the remnants that carry the bulk of cholesterol out to your tissues. But then we have the HDLs. These are your biological garbage trucks. They perform what we call reverse cholesterol transport, swooping through the bloodstream, picking up excess cholesterol from extrahepatic tissues, and hauling it all the way back to the liver for disposal.
So what happens when this intricate transportation network fails? Well, we get dyslipidemia. When these transport mechanisms break down or get totally overwhelmed by poor diet, genetics, or metabolic issues, it results in massive traffic jams of lipids in the bloodstream. For example, if you have a genetic defect in your LDL receptors, those LDL delivery trucks have literally nowhere to dock, and they just pile up in the plasma. This specific traffic jam is called type 2A hyperlipidemia, which you probably know as familial hypercholesterolemia.
To understand how to clear these traffic jams, we have to look at the body's natural cholesterol disposal mechanism, the enterohepatic circulation. It's a remarkably elegant recycling system. Step 1. The liver converts cholesterol into bile acids, which are essential for digesting dietary fats. Step 2. These bile acids are secreted into the small intestine. And step 3, in a supreme act of biological efficiency, the body reabsorbs about 85% of these bile salts right back into the bloodstream to be used again.
It's a continuous, closed-loop cycle between the liver, the gallbladder, and the small intestine. The liver constantly converts about half a gram of cholesterol to bile acids daily just to maintain this pool. Now, think like a clinician for a second. If we want to lower cholesterol in the blood,
What if we interrupt this recycling process? If we can stop the intestine from reabsorbing that 85% and instead force it to be excreted, the liver is going to panic. It will realize it's suddenly out of bile acids, and it will aggressively pull cholesterol straight out of the bloodstream to manufacture more. This exact mechanism is a massive target for both our dietary and pharmacological treatments.
Section 3. Assessing the risks.
Not all these cholesterol carriers behave the same way when it comes to the long-term health of our arteries. It all comes down to a molecular showdown. On one side, we have LDL. It carries up to 70% of our blood cholesterol. The real danger here is that these particles, especially the small, dense ones, can actually penetrate the inner lining of our blood vessels. Once they get trapped, they oxidize and form atherosclerotic plaques. That's why LDL is widely known as the atherogenic villain. On the other side, we have HDL, our anti-atherogenic hero. Because HDL actively clears cholesterol away from those arteries, high levels, specifically anything over 60 milligrams per deciliter, are incredibly protective against heart attacks and strokes. The ultimate goal in clinical lipid management? Shrink the villain and boost the hero.
Section 4. Dietary management strategies.
So knowing all this deep biochemistry, how can we practically manipulate these pathways using clinical nutrition? Well, food is quite literally information for your cells, and dietary management can be highly targeted. Take saturated fats, for example. They actually suppress your liver's LDL receptors, leaving more LDL stranded in your blood. Conversely, omega-3 and omega-6 polyunsaturated fatty acids can increase receptor activity, driving your LDL down. But perhaps the most elegant dietary intervention is viscous, water-soluble fiber, the kind found in oats and pectin. Remember our discussion on the enterohepatic circulation? These viscous fibers physically bind to bile salts in your gut, trapping them so they get excreted in your stool. This forces your liver to pull more cholesterol right out of your blood to replace the lost bile. It's a completely brilliant, natural way to lower your cholesterol.
And here is a number you should definitely commit to memory: 6 to 15%. That is the exact percentage you can reduce your LDL cholesterol simply by consuming 1.5 to 1.8 grams per day of plant sterols or stanols. These phytosterols, which you find in things like wheat germ, avocados, and fortified spreads, structurally mimic cholesterol. When you eat them, they literally compete with animal cholesterol for absorption in your intestine. Your body ends up absorbing the plant sterols and then aggressively secreting them back out, leaving the actual cholesterol behind to be safely flushed from your system.
Section 5. Medical and alternative treatments.
But what happens when diet and exercise just aren't cutting it? For patients with genetic conditions like familial hypercholesterolaemia, pharmacology isn't just an option, it's a necessity. Let's look at the heavy hitters. Statins are right at the top of the list. Famously discovered from fungi by researcher Akira Endo, statins work by completely blocking that exact HMG-CoA reductase enzyme we looked at in the biosynthesis pathway earlier. By stopping the liver from making its own cholesterol, the liver is forced to upregulate its LDL receptors, aggressively clearing LDL out of the bloodstream. We also have bile acid binding resins, which do synthetically exactly what oat fiber does naturally. And niacin is super unique because it's arguably the most effective drug we currently have for raising our protective HDL levels, sometimes by up to 30%.
But the scientific community is constantly searching for novel mechanisms, which brings us to alternative medicine. Take berberine, for instance, a bright yellow plant alkaloid with a long history in Ayurvedic and Chinese medicine. What's fascinating about berberine is that clinical studies show it can lower total cholesterol and triglycerides significantly, but it does it through an entirely different pathway than statins. Berberine actually increases the activity of a signaling protein called ERK, which ultimately tells the liver to form more LDL receptors on its surface. And it does this all without the muscle pain or side effects that some patients experience with statins.
As we wrap up this explainer, I want to leave you with a fascinating look into the future. Consider the ApoA1 Milano variant. It's a genetic mutation discovered in a small Italian village where people have dangerously low HDL and high triglycerides, yet their arteries remain perfectly clear and totally free of heart disease. Their mutant HDL acts like a superpowered plaque scraper. As we map out unique genetic mutations exactly like this one, we really have to ask ourselves, how will personalized medicine rewrite the rules of cardiovascular health? Will we soon be synthesizing superproteins to clear our arteries, rather than just blocking cholesterol production? The intersection of biochemistry and nutrition is evolving so rapidly, and understanding these fundamental mechanics is the very first step to unlocking true cardiovascular health. Thanks for joining me on this deep dive.